towards a structurally resolved human-protein interaction network. David F. Burke, Patrick Bryant, Inigo Barrio-Hernandez, Danish Memon, Gabriele Pozzati, Aditi Shenoy, Wensi, Alistair S. Dunham, Pascal Albanese, Andrew Keller, Richard A. Scheltema, James E. Bruce, Alexander Leitner, Petras Kundrotas, Pedro Beltrao, and Arne Elofsson. Nature Structural and Molecular Biology, Volume 30, pages 216-225, 2023. Cite this article. Abstract. Cellular functions are governed by molecular machines that assemble through protein-protein interactions. Their atomic details are critical to studying their molecular mechanisms. However, fewer than 5% of hundreds of thousands of human-protein interactions have been structurally characterized. Here we test the potential and limitations of recent progress in deep learning methods using AlphaFold 2 to predict structures for 65,484 human-protein interactions. We show that experiments can orthogonally confirm higher confidence models. We identify 3,137 high-confidence models, of which 1,371 have no homology to a known structure. We identify interface residues harboring disease mutations, suggesting potential mechanisms for pathogenic variants. Groups of interface phosphorylation sites show patterns of co-regulation across conditions, suggestive of coordinated tuning of multiple protein interactions as signaling responses. Finally, we provide examples of how the predicted binary complexes can be used to build larger assemblies, helping to expand our understanding of human cell biology. Similar content being viewed by others. Network-based prediction of protein interactions. Article Open Access. March 18, 2019. From Systems to Structure, Using Genetic Data to Model Protein Structures. Article January 10, 2022. Assessment of community efforts to advance network-based prediction of protein-protein interactions. Article Open Access. March 22, 2023. Main. Proteins are key cellular effectors determining most cellular processes. These rarely act in isolation, but instead, the coordination of the diversity of processes arises from the interaction among multiple proteins and other biomolecules. The characterization of protein-protein interactions, PPIs, is crucial for understanding which groups of proteins form functional units and underlies the study of the biology of the cell. Diverse experimental and computational approaches have been developed to determine the PPI network of the cell, that is, the interactome, with hundreds of thousands of human protein interactions determined to date 1, 2, 3. Protein interactions vary from transient interactions that regulate an enzyme to permanent interactions in molecular machines. The structural characterization of the human interactome has lagged behind, with experimental and homology models currently covering an estimated 15 protein interactions, 4, 5. The structural characterization of protein complexes is a critical step in understanding the mechanisms of protein function and in studying the impact of mutations, 4, 6, 7, 8, and the regulation of cellular processes via the post-translational tuning of binding affinities, 9, 10, 11, 12. Computational approaches for predicting the structures of interacting protein pairs are primarily based on identifying structural similarity for pairs of proteins against experimentally determined protein complexes, 4, 6, 13, 14. The Interactome 3D 4, 14 repository currently lists 7,625 predicted models based on homology of domains, a number similar to the 8,359 pairs listed having an experimentally determined model. In addition, co-evolution-based information has been used to predict protein interactions and to guide structural docking for bacterial proteins 15. Recently, neural network-based approaches have demonstrated the ability to accurately predict the structures of individual proteins 16, 17, and protein complexes 16, 18, 19, 20, 21. These approaches can correctly predict the structures of up to 60% of dimers 18 and have been used to predict structures of 1,506 Saccharomyces cerevisiae protein interactions 22. However, the application of these neural network models for the large-scale prediction of human complex structures has not been tested yet. Here, we assess the possibilities and limitations of applying AlphaFold 2 to modeling human protein interactions on a large scale. We predicted the complex structures for 2 sets of human interactions obtained using different experimental methods, comprising 65,484 unique human interactions. We show that it is possible to rank the models according to confidence, with 3,137 predicted structures ranked as highly confident. Further, we show that the higher confidence predictions are enriched among those supported by a combination of experimental methods. We showcase the value of a structurally resolved interactome by studying disease mutations and phosphorylation of interface residues. Finally, we provide some indication that binary complexes can be used to build higher-order assemblies. Structure prediction of human protein interactions. We selected experimentally identified human protein interactions from the Human Reference Interactome, HURI-2, and the Human Protein Complex Map, HU.map v.2.0. 3. HURI comprises protein interactions determined by yeast 2 hybrid Y2H screening 2, from which we modeled 55,586 pairs. From HU.map, we selected 10,207 high-quality Ps 3. While HURI is more likely to be enriched for direct protein interactions, including transient partners, the HU.map set is more likely to reflect stable protein interactions, including members of the same complex that may not be interacting directly. The overlap between the 2 datasets is small, 309 pairs, and a comparison with 2 large-scale compendiums of structural models 4 indicates that 62,019 of the combined pairs do not have experimental models, nor can they be modeled easily by homology, suggesting a large potential gain in structural knowledge. We predicted the structure of 65,484 non-redundant pairs using the fold-doc pipeline 18, based on AlphaFold 2, Ref 17. As in the fold-doc pipeline, we combined size and the predicted local distance difference test (PLDDT) scores of the interface into a single score to predict the doc score of a complex dubbed P-doc methods, which can rank models by confidence. We tested P-doc score by comparing the predicted models with 1,465 experimental models, of which 742, 50%, were correct, doc greater than 0.23. For predictions with P-doc greater than 0.23, 70%, 671 of 955, are well modeled, and for P-doc greater than 0.5, 80%, 521 of 651. We show in Figure 1a the distribution of P-doc for the predicted and random protein interactions, and provide data for all models in Supplementary Table 1. The P-doc of known interacting proteins tends to be higher than for the random set, with the predictions for HU.map showing on average higher confidence than the HURI set. Additionally, when selecting HU.map interactions, also supported by Y2H or cross-link data, cross-linking results in even higher confidence values, Figure 1a. This suggests that high-confidence models are enriched for protein interactions supported by the 2 types of methods associated with high affinity and direct interactions. We identified 3,137 structures, Figure 1b, as high-confidence models, P-doc greater than 0.5. The number of structures increased to 10,061 if a cutoff of 0.23 was used. Only 0.3% of the random set of models would be considered confident predictions at this cutoff. In Figure 1c, we show examples of predicted structures aligned to experimental or homology models, showing how the predictions and the confidence score relate to the observed alignments. For the majority of these cases, even with lower confidence values, the interaction interface is generally in good agreement, except for the interaction between subunits of the proteasome 26S complex, AT-PACE domain 2, PSMC2, and non-AT-PACE domain 11, PSMD11. It can be noted that several of the models in Figure 1c are parts of large complexes, PRDX2, PRDX3, members of the peroxidoxin family of antioxidant enzymes, RFC2, RFC5, subunits of heteropentameric replication factor C, RFC, UOB, parts of the 1433 family of proteins, tyrosine 3 monooxygenase / tryptophan 5 monooxygenase activation proteins beta, UOB, and gamma UOG, and RPL9, RPL18A, ribosomal proteins L9, RPL9, and L18A, RPL18A. This shows that AlphaFold 2 can predict the structures of directly interacting protein pairs present in large complexes. Figure 1. Application of AlphaFold 2 complex predictions to a large dataset of human PPIs. Figure 1. A. Distribution of model confidence score, PDOC, for predicted structures from two large human protein interaction datasets, Inu.map and WHO-RE, compared with confidence metrics from 2,000 random pairs of proteins. The WHO.map dataset was further subsetted to those that have support from Y2H, or cross-link data, cross-linking, or correspond to pairs with available experimental or homology modeling information, structure. B. Number of protein interactions with models built from both datasets and those that we consider being of high confidence, predicted, corresponding to those with PDOC greater than 0.5. C. Examples of predicted models, orange and green, overlapped with the corresponding experimental models, gray, and the observed DOC or predicted PDOC quality of the models. Source data. Full-size image. Features impacting prediction confidence. As shown in Figure 1a, protein pairs present in the Protein Data Bank, PDB, are enriched in high-scoring models compared with pairs in WHO-RE and WHO.map. There could exist several possible explanations for this, such as the inability of AlphaFold 2 to identify transient or indirect interactions. Nevertheless, it is also possible that the two high-throughput datasets contain non-interacting pairs. Therefore, to understand this difference better, we first studied an additional dataset created from large, greater than 10 chains, heteromeric protein complexes. The set of large complexes consists of 12 large heteromeric protein complexes, and all non-identical pairs of protein chains in each complex were docked with each other. These pairs can be divided into the ones with direct interaction and those that do not interact directly. Here, we used a definition of more than 20 contacts of less than 8 a ring between alphas to exclude small interaction interfaces. When a complex contained multiple copies of identical chains, all interactions were included to allow for alternative interactions between the chains. The difference in PDOC scores between the direct and indirect interacting pairs is striking, where only 6% of the indirect pairs have a PDOC score greater than 0.5 compared with 38% of the directly interacting pairs, Figure 2a. This shows that directly interacting pairs often can be predicted even when they are part of large complexes, in contrast to indirectly interacting pairs. Figure 2. Protein and interaction features impacting on prediction confidence, analysis of different datasets, Figure 2. In all subfigures, proteins from WHO-RE in green, WHO.map gray, quorum orange, and from large PDB complexes blue. A. PDOC values of directly and indirectly interacting proteins from the same complex, blue. For comparison, the WHO-RE and WHO.map data are shown with thin lines. B. PDOC values of quorum, orange, WHO-RE, green, and WHO.map, gray datasets. C. Fraction of residues predicted to be disordered, PODDT less than 0.5, shows that protein pairs in WHO-RE are enriched in disorder. D. Proteins in WHO-RE have fewer sequences in the paired MSAs as measured by the mean number of efficient sequences in the MSA MEF. E. Proteins that share subcellular localization, solid lines, are enriched in high PDOC scores in all three datasets. F. Only protein pairs in WHO.map are co-expressed according to string, using similarity in genotype tissue expression, GTEx, and co-expressed pairs are enriched in pairs with high PDOC scores. Source data. Full-size image. WHO.map has many more high-confidence predictions than WHO-RE, which is based on Y2H experiments. To further understand this difference, we first analyzed a subset of all protein pairs from the Quorum 23 database, the best manually curated database of mammalian protein complexes, and predicted the interaction of all pairs in the same complex. The average PDOC score of Quorum is slightly higher than for WHO.map, but the number of high-quality predictions is similar, 16% versus 19%, indicating that the different databases of protein complexes have a similar fraction of high-confidence predictions and that Hu-Ri is the outlier, Figure 2b. It is unlikely that the Y2H in Hu-Ri data should contain a large set of indirect interactions, as only 2 human proteins are expressed in the same cell. Therefore, there must be another reason for the few high-confidence predictions. We examine the properties of the pairs present in the 2 datasets. Here, it can be seen that Hu-Ri proteins differ from the Hu-Dot map and other datasets in 2 ways. Hu-Ri protein pairs contain more intrinsic disorder, Figure 2c, and have fewer efficient sequences, MEF, in their multiple sequence alignments, MSAs, Figure 2d. In these figures, it can also be seen that the p-doc values tend to increase with less disorder and more sequences in the alignments, although it is clearly not an absolute relationship. Further, protein pairs in Hu-Ri are less likely to be found in the same subcellular compartment, Figure 2e, and have similar co-expression profiles, Figure 2f. Considering all this, it is likely that many protein interactions in Hu-Ri are transient, and that AlphaFold 2 cannot reliably predict such interactions. Cross-linking support for predicted complex structures. Chemical cross-linking followed by mass spectrometry is an approach which can be used to identify reactive residues, usually lysines, that are in proximity, as constrained by the geometry of the cross-link agent used. The identification of such residues across a pair of proteins can help define the likely protein interface. To determine if the predicted complex structures agree with such orthogonal spatial constraints, we obtained a compilation of crosslinks for pairs of residues across 528 protein pairs with predicted models, Figure 3a, Supplementary Table 1, and methods. In total, 51% of the models had 1 or more crosslinks at a distance below the expected maximal distance possible, Figure 3a. Restricting the predicted models to higher confidence by the p-doc score increased the fraction of complexes with acceptable crosslinks, reaching 75% for p-doc scores greater than 0.5, Figure 3a. This result is in line with the benchmark results above. Figure 3. Crosslink support for predicted complex models. Figure 3a. The numbers and ratios of predicted structures having crosslink information for pairs of residues that bridge the 2 proteins in the predicted structure, broken down by the crosslinks that satisfy their expected maximal distance, and by the predicted quality of the model, p-doc. B. Examples of predicted structures of high confidence, with no previous structural information and supported by at least 1 crosslink, indicated with blue line, Erlin 1/Erlin 2 complex, B. IMMT and CHCHD3, components of the mitochondrial inner membrane Michels complex, C. The complex of transfer RNA guanine in 7-methyltransferase, metal, with its non-catalytic subunit, WDR4, D, and the heterogeneous nuclear ribonucleoprotein C, HNRNPC, and the RNA-binding protein, Rayleigh, E. Source data. Full-size image. In total, we have identified 479 crosslinks providing supporting evidence for 171 predicted complex structures with p-doc greater than 0.5. Of these, 41 correspond to complex structures with no experimental structure or homology models, from which we selected some to illustrate, Figure 3be. Figure 3b shows the AlphaFold 2 AF2 model for the full length of the Erlin 1/Erlin 2 complex, which mediates the endoplasmic reticulum-associated degradation, ERAD, of inositol 1,4,5 trisphosphate receptors, IPRs. AlphaFold 2 predicts a globular domain, 1 to 190, followed by an extended helical region with a kink around amino acid position 280. Unlike the model in Interactome 3D, the paralogous proteins are stacked side by side with the hydrophobic face of the helices, buried, and the hydrophilic face, mainly Lys, exposed to solvent. A crosslink between the C-terminal residues, K275, Erlin 1, and K287 is predicted to bridge a distance of 18A ring, supporting the predicted model. In Figure 3c, we show the model for proteins IMMT and CHCHD3, components of the mitochondrial inner membrane Michels complex. AlphaFold 2 predicts a globular helical domain at the C-terminal end of IMMT, 550 to 750, to interact with the C-terminal end of CHCHD3, 150 to 225. This is supported by data of 3 crosslinks between K173, CHCD3, and K565, IMMT, and K203, CHCD3, to both K714 and K726 of IMMT. Figure 3D shows the complex of transfer RNA guanine N7 methyltransferase, metal, with its non-catalytic subunit, WDR4. The structure of WDR4 has not yet been solved experimentally, but contains WD40 repeats, which are expected to form a beta propeller domain, as predicted here. The metal domain is predicted to interact with the side of the WDR40, away from the ligand-binding pore. This orientation is supported by a crosslink between K122, WDR4, and K143, metal, 18A ring. Finally, in Figure 3E, we show the predicted complex structure for the heterogeneous nuclear ribonucleoprotein C, HNRNPC, and the RNA-binding protein, Rayleigh. 2 regions in both proteins are predicted with high confidence, PLDDT greater than 70, with the lower confidence regions not shown. The N-terminal domain in HNRNPC, 16 to 85, is predicted to interact with the N-terminal domain of Rayleigh, 1 to 100. A long helix in HNRNPC, 185 to 233, is predicted to interact with a helix in Rayleigh, 169 to 228. This interhelix interface is supported by cross-linking data for 3 pairs of lysines at either end of the helices, 189, 222, 229, 179, and 232, 183. Disease-associated missense mutations at interfaces. Missense mutations associated with human diseases can alter protein function via diverse mechanisms, including disrupting protein stability, allosterically modulating enzyme activity, and altering PPIs. Structural models can allow the rationalization of possible mechanisms of interface disease mutations. To determine the usefulness of the predicted structures, we compiled a set of mutations located at interface residues that were previously experimentally tested for the impact on the corresponding interaction 24. We then performed in silico predictions of changes in binding affinity upon mutations using Folds 25 and observed that mutations known to disrupt the interactions are predicted to have a strong destabilization of binding compared with mutations known not to have an effect. Figure 4a and Supplementary Table 2. Very high confidence, PLDDT greater than 90, of the mutated residues led to more substantial discrimination between mutations known and not known to disrupt the complex formation, Figure 4a, indicating that only very accurate models are useful when using the Folks force field for estimating the impact of binding affinity of mutations. Figure 4. Disease mutations at protein complex interface residues. Figure 4a. Box plot showing the distribution of changes in predicted binding affinity, delta delta G, for mutations known to have an impact, orange, versus the ones with neutral effect, green. The boxes represent the first and third quartiles. The upper whisker extends from the third quartile to the largest value no further than 1.5 times interquartile range, IQR. The lower whisker extends from the first quartile to the smallest value at most, 1.5 times IQR. The statistical significance of the differences was tested using 2-sided Wilcoxon rank sum test with continuity correction. The total number of data samples was 1,320, 900 neutral, 420 impact, Supplementary Table 2. The mean, and max values for the subsets are as follows. Experimental neutral, minus 0.5, 1.0, 5.1. Experimental impact, 0.1, 3.4, 8.6. AFPODDT greater than 90 neutral, minus 0.9, 0.3, 4.7. AFPODDT greater than 90 impact, minus 2.7, 1.0, 12.5. AF70 less than PODDT greater than 90 neutral, minus 2.0, 0.3, 9.0. AF70 less than PODDT greater than 90 impact, minus 4.0, 0.5, 7.0. AF50 less than PODDT greater than 70 neutral, minus 1.2, 0.1, 2.7. AF50 less than PODDT greater than 70 impact, minus 0.2, 0.3, 1.7. AF-PODDT less than 50 neutral, minus 2.6, 0.1, 2.0. AF-PODDT less than 50 impact, minus 2.6, minus 0.3, 0.3. B. Unique PPI pairs for high confidence models, P-Doc greater than 0.5 in total, with mutations in cancer mapped to the interface, all in top 25% ratios, and with pathogenic or likely pathogenic clinical variants mapped to the interface. C. Modules related to relevant biological processes. The color of the edge represents the presence of cancer mutations in the interface, top 25% ratio, color red, and the shape, the presence of pathogenic clinical variants, double line. DG. Selected relevant structures with no previous structural knowledge showing clinical variants or mutations in cancer mapped to the interface, mutated residues in red. The interface of WDR4 metal 1D, the interface of ULDOC1E, the interface of TWIST1F, and the interface of RAD51DG. Source data. Full-size image. Next, we mapped human disease from ClinVar and cancer mutations from the Cancer Genome Atlas to the interface residues defined by the set of high-confidence protein complex predictions, P-Doc greater than 0.5, Supplementary Table 1. The WHO.map and WHO-RE confident predictions identified 280 interfaces carrying pathogenic mutations and 602 interfaces corresponding to the top 25% of recurrently mutated interfaces in cancer, defined as the highest number of mutations per interface position, Figure 4b, and methods. We find a strong enrichment in pathogenic versus benign mutations at interface residues relative to the rest of the protein, 2.3-fold enrichment, p-value 2.7 times 10^-31. We illustrate in Figure 4c examples of protein network clusters with interface disease mutations across a range of biological functions. For example, interface mutations in chromatin remodeling, including members of SWI/SNF complex, SMARCD1, SMARCD2, SMARCD3, and several transcription factors related to development, for example, TCF3, TCF4, LMO1, and LMO2. We selected examples of interfaces with disease mutations and no previous experimental data or homology to available models, Figure 4DG. Figure 4D shows the interface of WDR4 metal 1, which has supporting crosslink information described above. WDR4 has two annotated pathogenic variants at this interface, linked with Gallo-Emoid syndrome 6, with the highlighted R170 participating in interactions with a negatively charged residue of metal 1. Figure 4E shows an example of an interface with 32 recorded interface mutations in cancer for both proteins, including the highlighted arginines in ULDOC1, which form electrostatic interactions with the opposite chain. TWIST1 has several annotated pathogenic mutations, including L149R and L159H, which are at residues buried in the interface, Figure 4F. In particular, the L149R mutation, associated with Sayre-Richardson syndrome, would strongly disrupt packing. The R118G mutation would disrupt the interaction with residue F22 main chain O in TCF4. In RAD51D, we found the mutation R266C, breast ovarian cancer, familial, which interacts across the interface with XRCC2, Figure 4 grams, and paralogous genes involved in the repair of DNA double-strand breaks by homologous recombination. Interestingly, we also found mutations at R239 to TRP/GLN/GLY associated with breast ovarian cancer, which interacts with TYR119 in XRCC2, which itself is also annotated as having mutations linked to hereditary cancer predisposing syndrome. Phosphorylation of protein complex interfaces. Protein phosphorylation can regulate protein interactions by modulating the binding affinity via the change in size and charge of the modified residue, Over 100,000 experimental human phosphorylation sites have been determined to date 26,27, but only 5 to 10% of these have a known function 28. Mapping phosphorylation site positions to protein interfaces can generate mechanistic hypotheses for their functional roles in controlling protein interactions. We used a recent characterization of the human phosphoproteome 26 to identify 4,145 unique phosphosites at interface residues among the highly confident models. The average functional importance, defined by the functional score described earlier 26, was generally higher than random for phosphorylation sites at interfaces, figure 5a, and we found some enrichment for targets of multiple kinases, including tyrosine kinases, ERBB2, AXL, ABL2. Fair. Figure 5b. This suggests that some interfaces may be under coordinated regulation by specific kinases and conditions. Figure 5. Co-regulation of phosphorylation sites at interface residues. Figure 5. A. Distribution of phosphosit functional scores for phosphosites at interface residues and random phosphosites. The min, mean, and max values were as follows. Random equals 0.02, 0.26, 0.98. Hurike equals 0.06, 0.37, 0.99. Hu.map equals 0.06, 0.33, 0.99. The boxes represent the first and third quartiles. The upper whisker extends from the third quartile to the largest value no further than 1.5 times IQR. The lower whisker extends from the first quartile to the smallest value at most, 1.5 times IQR. B. Enrichment of kinase substrates among phosphosites at interface residues. The p-value was derived from an overrepresentation analysis using a one-sided hypergeometric test, n equals 7,150. C. Hierarchical clustering of the pairwise correlation values for changes in phosphosit levels across conditions. Groups of phosphosites showing high correlation values were defined as clusters, 1 to 16, as indicated in colors along the outside of the clustergram. D. Degree of regulation of phosphosites from each cluster in a select panel of conditions, defined by a one-sided z-test comparing the fold change of the phosphosites in a cluster, compared with the entire distribution of fold changes in that condition. The result is summarized as the minus log p-value, and signed as positive if the median value is above the background or negative otherwise. E. Gene ontology enrichment analysis for the proteins with phosphosites annotated to select clusters. Source data. Full-size image. To identify potentially co-regulated interfaces, we collected measurements of changes in phosphorylation levels across a large panel of over 200 conditions 29. We retained 260 phosphosites that had a significant regulation in 3 conditions, and then computed all-by-all pairwise correlations in phosphosit fold changes across conditions, Supplementary Table 1. We clustered these phosphosites by their profile of correlations, Figure 5c, identifying 16 groups of co-regulated interface phosphorylation sites, Figure 5c, and Supplementary Table 3. For each group of phosphosites, we identified the conditions where these have the strongest up or down regulation, Supplementary Figure 1, and plotted a subset of conditions in Figure 5d. We also performed a gene ontology enrichment analysis for each group of co-regulated phosphosites, including both proteins of the modified interfaces, to search for common biological functions, Figure 5e and Supplementary Table 4. Here, one-sided hypergeometric tests were used for statistical analysis. For example, we observed a cluster of interface phosphosites in proteins related to intermediate filaments, Cluster 7, which show strong regulation patterns along the cell cycle, down-regulated in S phase and up-regulated in G1 and mitosis. Phosphosites in Cluster 1, cell cycle G1 S phase transition, show the opposite trends, with up-regulation in late S phase and down-regulation in G1 and mitosis. Some clusters show regulation under specific kinase inhibition, which may provide novel hypotheses for kinase regulation of specific processes. For example, phosphosites in Cluster 9, regulation of chromosome assembly, tend to be upregulated after inhibition of ROCK and upregulated after inhibition of mTOR. While not all phosphosites at interfaces are likely to regulate the binding affinity, This analysis provides hypotheses for the potentially coordinated regulation of multiple proteins by tuning of their interactions after specific perturbations. Higher-order assemblies from binary protein interactions. Proteins interact with multiple partners either simultaneously, as part of larger protein complexes, or separated in time and space. This is also reflected in our structurally characterized network, where proteins can be found in groups as illustrated in a global network view of the protein interactions with confident models, Figure 6, Supplementary Figure 2, and Supplementary Data 1. One key benefit of structurally characterizing an interaction network is the identification of shared interfaces for multiple interactors. As an example, we highlight GDI1, Rab-GDP dissociation inhibitor alpha, which interacts with multiple Rab proteins, regulating their activity by inhibiting the dissociation of GDP. The predicted complex structures for these interactions show how these share the same interface and therefore cannot co-occur. Other clusters in the network suggest that the proteins form larger protein complex assemblies with many-to-many interactions. As the use of AlphaFold 2 for predicting larger complex assemblies can be limited by computational requirements, we tested whether the structures for pairs of proteins could be iteratively structurally aligned. We tested this procedure on a small set of complexes covered in this network, with known structures and the number of subunits ranging from 5 RFC complex to 5 core complex to 14 20S proteasome. We then aligned an experimentally determined structure with the predicted models, Figure 6, Gray experimental model. These examples showcase the potential and also limitations of this procedure. Figure 6, Protein complex predictions for higher-order assemblies. Figure 6. The middle circle is a network view of all PPIs predicted with high confidence, p-doc greater than 0.5. The edges and nodes are colored in red if there is a previous experimental or homology model for the interaction, and blue if such information is unavailable. We selected 4 examples of recapitulated complexes, yellow circles and black arrows, plotted in further detail. In these small networks, only the edges are colored based on structural evidence. In the case of Rab-GDP, the faded nodes and edges represent predictions with slightly lower confidence, p-doc greater than 0.3. Full-size image. The core complex is composed of 5 subunits with 1-to-1 stoichiometry. All subunits can be modeled, with the final complex generally agreeing, Figure 6, with the cryo-M structure for these subunits, PDB:6 NMI. The most significant difference to the cryo-M model is the relative positioning of the ERCC3 subunit. The exact final model obtained can vary depending on the aligned pairs, with multiple possible final conformations, Supplementary Figure 3. Figure 6 illustrates the conformation that best matches the cryo-M model in PDB:6 NMI. For example, for the T5 core complex, there is a predicted model where the complex adopts a more open conformation, as seen in PDB:5 OQJ, and alternative predicted placements of the GTF2H1 subunit. The RFC complex is also composed of 5 subunits with 1-to-1 stoichiometry. One iterative alignment of pairwise protein interactions builds a model that includes all 5 subunits organized similarly to that observed in the PDB:6 VVO cryo-M structure, Figure 6. In this predicted model, the subunits RFC 2/5/4/3 match the experimentally observed model well, but there are apparent deviations introduced by compounding errors in alignment by this iterative process. Individual subunits in the cryo-M structure can be aligned to each of the model subunits well, but then the alignment of the rest of the model is progressively worse the further away the subunits are positioned from the aligned subunit. The RFC1 subunit is individually not well predicted. Further, the RFC3-RFC5 interaction pair is predicted with high confidence, while, in fact, these do not share a direct contact in the experimental structure. AlphaFold 2 places RFC3 at the RFC5-RFC4 interface, likely due to the structural similarity between RFC3 and RFC4. Encouraged by the examples tested, we defined an automatic procedure to generate larger models by iterative alignment of pairs, methods. We start building all possible dimers in a complex, then sort them by p-doc, and start building from the first-ranked dimers. Next, we add the highest-ranked dimer, which shares one subunit with the complex if it does not overlap. This is repeated for all dimers until the complex is complete or no additional proteins can be added. We tested this on the 20th proteasome, a particularly challenging example, with stoichiometries different from 1-to-1 and homologous subunits. This automatic procedure could build a model containing all 14 subunits, half of the proteasome, which are mostly placed in agreement within the experimental model, figure 6. However, the exact order of the chains is incorrect, that is, at each location an incorrect protein is placed, highlighting that AF2 cannot distinguish which two proteins interact from a set of homologous proteins. Two additional proteins where we could build a good model are heterodisulfide reductase from Methanothermococcus thermolithotrophicus, PDB:5ODC, and the eukaryotic translation initiation factor 2B from Schizosaccharomyces pombe, PDB:5BO4, supplementary figure 4. For PDB:5ODC, we could build a complete model of the protein with an RMS deviation of 6.0 A ring, TM score 0.90, 30, starting from dimers. However, for PDB:5BO4, it was not possible as the chains started overlapping when we tried to build a larger model. However, if we build trimers and then use all three dimers from these trimers, we can build a complete model with an RMS deviation of 7.3 A ring, TM score 0.86, showing that it is sometimes necessary to use larger subunits to assemble the complexes. Results from a follow-up study 31 show that it is often possible to build the structures of complexes if the subunits are well predicted. In summary, we find that it is possible to iteratively align structures of pairs of interacting proteins to build larger assemblies, but we also identified issues that limit this procedure at the moment. Concluding discussion. We have predicted complex structures for pairs of human proteins known to physically interact from two different datasets based on different experimental approaches. We note that the source of data used for the protein interactions is important and impacts the fraction of models that can be confidently predicted. Our analysis suggests that protein interactions supported by a combination of affinity, cofraction, and complementation-based methods result in higher confidence models. We believe these protein interactions tend to correspond to high-affinity interactions, which are very likely to share a direct physical permanent interaction. We show that it is possible to use metrics from the models, for example, p-doc score, to rank higher confidence models, providing an additional accuracy level to large-scale PPI studies, and in the future to provide additional high-quality targets for detailed studies of stable complexes. Experimental data from cross-link mass spectrometry experiments provide an ideal resource for further validating these predictions via orthogonal means. Based on comparisons with solved structures, we suggest that models with p-doc greater than 0.5 are 80% likely to be correct. Additionally, models with lower scores, p-doc greater than 0.23, are still 70% likely to contain many correct solutions and may highlight correct interfaces. Such lower confidence models are likely to be useful for generating hypotheses and large-scale analyses of global properties. Equally important is the caveat that high confidence predictions will still contain errors, and, in particular, we note that in protein complexes containing paralogous proteins, which is common in higher eukaryotes 32, the current procedure cannot identify the exact pairing of the protein. For such cases, additional methods need to be developed. Structural models for protein interfaces are critical for understanding molecular mechanisms and the impact of mutations and post-translational modifications. We illustrate this using disease mutations and phosphorylation data. While much disease-associated variation is often found in non-coding regions of the genome, the growth of exome sequencing of large cohorts of patients will lead to discovering many more protein mutations linked to disease, which will require such large structural characteristics. Both for mutations and for phosphorylation sites, we think these analyses should be seen as generating hypotheses for further testing, and we make this information available in the supplementary material to facilitate such future work. Finally, we show that it is in principle possible to build structural models for larger assemblies from predicted binary complexes. In a follow-up paper, we have shown that it is possible to build large assemblies fully automatically by using predictions of dimers and trimers 31. Aspects that may limit this include the structural homology between subunits, unknown subunit stoichiometries, and limits in the predicted interactions 31. Additional work will be needed to determine the exact stoichiometry and to design methods and score systems to build such larger complex assemblies, as well as to predict the interactions of proteins with weak and transient interactions. Methods: protein interaction data and annotations. Human protein pairs known to physically interact were obtained from the WHO.map dataset, retaining pairwise protein interactions with greater than or equal to 0.5 confidence, and most interactions from the WHO-RE dataset. These interactions were further enriched by obtaining annotations on cross-linked peptides matched across pairs of interaction proteins, disease-related mutations, and protein phosphorylation sites in the selected proteins. In addition, all non-homologous pairs from 12 protein complexes, Supplementary Table 5, and 4,320 protein pairs from 2,102 different protein complexes, Supplementary Table 6, in Quorum 23, were used for additional analyses. A complete list of all datasets is available from the supplementary data. A subset of crosslink data was collected from refs 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, and filtered for peptides unique to only one protein sequence. A crosslink was considered validated by the structure if the distance between the epsilon amino groups on the side chains of the relevant pair of lysine residues was within 32 a ring. Clinical missense variants associated with disease were collected from ClinVar. We selected only those having pathogenic or likely pathogenic effects, which were mapped to uniprot protein sequences using VAR-map. The final list of mutated positions was then compared with the interface positions. We obtained a list of protein phosphorylation sites with predicted functional relevance 26, phosphosid annotations 28, and regulation of phosphorylation sites across a large panel of conditions 29. These phosphosids were also mapped to interface positions as defined by the predicted models. All protein interaction networks were processed using R packages iGraph v.1.2.5 and cgraph v.1.9. and further graphical editing was done using Cytoscape 44. Protein complex prediction. To predict protein complexes of pairwise interactions, we used the fold-doc pipeline 18 based on AlphaFold 2, Ref 17. We used the option of fused plus paired MSAs and ran the model configuration M1101 as this provides the highest success rate accompanied by a 20-fold speedup. Both the fused and paired MSAs were constructed by running hublets on every single chain against Uniclus 30. The fused MSA was generated by simply concatenating the output of each of the single-chain hublets runs for two interacting chains. The paired MSA was constructed by combining the top hit for each matching OX identifier between two interacting chains, using the output from the single-chain hublets runs. P-Doc confidence score. To score models, we used features from the predicted complexes to calculate the predicted DOC score, P-Doc. This score is defined with the following sigmoidal equation, where x equals average interface PLDDT log number of interface contacts. The parameters were optimized to predict the DOC score using the dataset from Ref 45. The number of interface contacts is defined as elsewhere in this paper, any residues with an interface atom within 10 a ring of the other chain, and the PLDDT is the predicted LDDT score from AlphaFold 2 taken over the interface residues as defined by the interface contacts. Building larger complexes from binary protein interactions. A simple procedure to build larger complexes from a set of paired models was developed. All dimers in the set are by default ranked by their P-Doc values. 1. The building is started from a single dimer, by default the dimer with the highest P-Doc value. This is referred to as the complex. 2. All other dimers in the set are then tried to be added to the complex. Starting with the one with the second highest P-Doc, A chain is added to the complex if exactly one chain of the dimer is identical to one chain in the complex. B. The structure of these two chains is similar enough, default TM score greater than 0.8. C. The dimer is then rotated so that the two chains overlap. D. The second chain in the dimer does not clash with more than 25% of its residues, C alpha distance less than 5A ring, with any chain in the complex. 3. If a chain is added, the procedure is started over again and repeated until no more chains can be added. Analysis of phosphosids in the protein-protein interfaces. Phosphosid residues in interfaces were identified from a previously published comprehensive list of known human phosphosids 26. Kinases associated with phosphorylation of interface residues were obtained from the Phosphosid Plus database. An overrepresentation analysis of kinases was performed using a hypergeometric test. Highly regulated interface phosphosids were defined as those with more than twofold change in phosphorylation in more than two perturbation conditions across a collated phosphoproteomics dataset comprising a range of physiological conditions and drug treatments. 29. Pearson correlation was calculated amongst these regulated phosphosids, and clusters of co-regulated phosphosids were identified using hierarchical clustering, Ward method, of Euclidean distances of the correlation matrix. Phosphosid clusters were created by cutting the dendrogram at the appropriate level using the Q-tree H equals 17 function in R. Phosphosid clusters that were significantly regulated in each perturbation condition were identified by a Z-test from the comparison of fold changes in phosphosid measurements of all phosphosids in a cluster against the overall distribution of phosphorylation fold changes across the condition. Gene ontology overrepresentation of each cluster was performed separately using a hypergeometric test in R. The gene ontology terms were obtained from the C5 category of the molecular signature database, Zig of 7.1, 46. All overrepresentation analyses were performed using the enricher function of the cluster profiler package, v.3.12.06 in R. Comparison with other databases. All proteins used here were mapped to UniProt47 to retrieve subcellular localization, string 48 for co-expression and other interaction data, and GTEX49 for tissue-specific expression. Reporting summary. Further information on research design is available in the Nature Portfolio Reporting Summary linked to this article. Data availability. All datasets and metadata are available from https://doi.org/10.17044/siliflab.168666202.v1. Further, all models generated as well as some of the multiple sequence alignments can be found at https://archive.bioinfo.say/Huentov2/. Source data are provided with this paper. Code availability. All code used in this project can be found at https://gitlab.com/elofssonlab/Huentov2/. Tools to run AlphaFold 2 for combined folding and docking can be found at https://gitlab.com/elofssonlab/folddoc/. References. Orchard, S. et al. The MINT ACT project, INT ACT as a common curation platform for 11 molecular interaction databases, nucleic acids Res42, D358, D363, 2014. Luck, K. et al. A reference map of the human binary protein interactome. Nature 580, 402-408, 2020. Drew, K., Wallingford, J.B. and Marcotte, E.M. Who.map 2.0. Integration of over 15,000 proteomic experiments builds a global compendium of human multi-protein assemblies. Mol Cystabio 17, E10016, 2021. Mosca, R., Kjol, A. and Aloy P. Interactome 3D, adding structural details to protein networks. NATA Methods 10, 47-53, 2012. Burley, S.K. et al. RCSB Protein Data Bank, powerful new tools for exploring 3D structures of biological macromolecules for basic and applied research and education in fundamental biology, biomedicine, biotechnology, Bioengineering and Energy Sciences. Nucleic Acids, Res 49, D437, D451, 2021. Wang, X et al. Three-dimensional reconstruction of protein networks provides insight into human genetic disease. Nat Biotechno 30, 159 to 164, 2012. Kombarov, A. et al. Comprehensive Assessment of Cancer Missense Mutation Clustering in Protein Structures. Proc National ICATS I USA 112, E5486, E5495, 2015. Porta Pardo E. Garcia Alonso L. Harab T. Dopazzo J. and Godzik A. A Pan-Cancer Catalog of Cancer Driver Protein Interaction Interfaces Closed Compute Bio 11, E1004518, 2015. Beltrao, P. et al. Systematic Functional Prioritization of Protein Post-Translational Modifications. Cell 150, 413 to 425, 2012. Nishi H., Hashimoto K. and Panchenko A.R. Phosphorylation in Protein-Protein Binding. Effect on Stability and Function. Structure 19, 1807 to 1815, 2011. Sosteric, N. et al. Effects of acetylation and phosphorylation on subunit interactions in three large eukaryotic complexes. Mole Cell Proteome 17, 2387 to 2401, 2018. Betts, M.J. et al. Systematic Identification of Phosphorylation-Mediated Protein Interaction Switches. The Closed Compute Bio 13, E1005462, 2017. Zhang, Q.C. et al. Structure-Based Prediction of Protein-Protein Interactions on a Genome-Wide Scale. Nature 490, 556 to 560, 2012. Mosca R. Kjol A. Stein A. Olivella R. and Aloy P3 did a catalog of domain-based interactions of known three-dimensional structure. Nucleic Acids Res 42, D374, D379, 2014. Kong Q. Anishchenko I. Ovchennikov S. and Baker D. Protein Interaction Networks Revealed by Proteome Coevolution. Science 365, 185 to 189, 2019. Beck, M. et al. Accurate Prediction of Protein Structures and Interactions Using a Three-Track Neural Network. Science 373, 871 to 876, 2021. Jumper, J. et al. Highly Accurate Protein Structure Prediction with AlphaFold. Nature 596, 583 to 589, 2021. Bryant P. Pozzati G. and Elofsson A. Improved Prediction of Protein-Protein Interactions Using AlphaFold 2. Nat Command 13, 1265, 2022. Pozzati, G. et al. Limits and Potential of Combined Folding and Docking Using Pecans Dock. Bioinformatics 38, 954 to 961, 2022. Actal, M. et al. A Structural Biology Community Assessment of AlphaFold 2 Applications. NatStruct Mol Bio 29, 1056 to 1067, 2022. Evans, R. et al. Protein Complex Prediction with AlphaFold Multimer. Preprint at BioRxiv https://doi.org/10.1101/2021.10.04.46303 2021. Humphreys, I.R. et al. Computed Structures of Core Eukaryotic Protein Complexes. Science 374, and Eben 4805, 2021. Georgiou, M. et al. Quorum. The Comprehensive Resource of Mammalian Protein Complexes, 2019. Nucleic Acids Res 47, D559, D563, 2019. IMEX Consortium Curators et al. Capturing Variation Impact on Molecular Interactions in the IMEX Consortium Mutations Dataset, Nat Command 10, 2019. Delgado, J., Radusky, L.G., Shinferoni, D. and Serrano, L. Folks 5.0, Working with RNA, Small Molecules and a New Graphical Interface. Bioinformatics 35, 4168 to 4169, 2019. Ochoa, D. et al. The Functional Landscape of the Human Phosphoproteome, Nat Biotechno 38, 365-373, 2020. Lawrence, R.T., Searle, B.C., Lovett, A. and V.N. J. Plug-and-play analysis of the human phosphoproteome by targeted high-resolution mass spectrometry. Nat Methods 13, 431-434, 2016. Hornbeck, P.V. et al. Phosphocyte Plus, 2014. Mutations, PTMs and recalibrations. Nucleic acids Res 43, D512, D520, 2015. Ochoa, D. et al. An Atlas of Human Kinase Regulation. Mol Cyst Bio 12, 888, 2016. Zhang Y. and Skolnick J. Scoring Function for Automated Assessment of Protein Structure Template Quality. Proteins 57, 702-710, 2004. Bryant, P. et al. Predicting the Structure of Large Protein Complexes Using AlphaFold and Sequential Assembly. Nat Common 13, 6027, 2022. Marchant, A. et al. The Role of Structural Pleiotropy and Regulatory Evolution in the Retention of Heteromers of Paralogs. eLife 8, E46754, 2019. Ugandar, K. et al., Max Linker. Proteome-wide cross-link identifications with high specificity and sensitivity. Mole Cell Proteome 19, 554-568, 2020. Schwepp, D.K. et al. Zlinkov 2.0, Integrated Large-Scale Structural Analysis of Protein Cross-Linking Data. Bioinformatics 32, 2716-2718, 2016. Klikov, O., Vandersvan, C., Heck, A.J.R., Meyer, A.B., and Scheltema, R.A. Missing regions within the molecular architecture of human fibrin clots structurally resolved by XLMS and integrative structural modeling. Proc National ICAD, SI USA 117, 1976-1987, 2020. Steigenberger B., Peters, R.J., Heck, A.J.R., and Scheltema, R.A. Fox. An IMAC enrichable cross-linking reagent, ACS Sent Sci 5, 1514-1522, 2019. Klikov, O. et al. Efficient and Robust Proteome-Wide Approaches for Cross-Linking Mass Spectrometry. Nat Prota 13, 2964-2990, 2018. Fasci D., Van Ingen H., Scheltema, R.A. and Heck, A.J.R. Histone interaction landscapes visualized by cross-linking mass spectrometry in intact cell nuclei. Mole Cell Proteome 17, 2018-2033, 2018. Yeliseyev, B. et al. Structure of a human cap-dependent 48S translation pre-initiation complex. Nucleic acids Res 46, 2678-2689, 2018. Gustout, D. et al. The chaperone and trick/CCT associates with prefoldin through a conserved electrostatic interface essential for cellular proteostasis. Cell 177, 751-765.E15, 2019. Klatt, F. et al. A precisely positioned MED12 activation helix stimulates CDK8 kinase activity. Proc National ICATSI USA 117, 2894-2905, 2020. Sabbath, K. et al. INTS 10, INTS 13, INTS 14 form a functional module of integrator that binds nucleic acids and the cleavage module. Nat Command 11, 3422, 2020. Muhammad, W.I. et al. The human GID complex engages two independent modules for substrate recruitment. MBO Rep 22, E52981, 2021. Shannon, P. et al. Cytoscape, a software environment for integrated models of biomolecular interaction networks. Genome Res 13, 2498-2504, 2003. Green, A.G. et al. Large-scale discovery of protein interactions at residue resolution using coevolution calculated from genomic sequences. Nat Command 12, 1396, 2021. Subramaniam V. Vincent I.R. and Jodi, S. Upregulation and dephosphorylation of cofilin, modulation by CD44 variant isoform in human colon cancer cells. Examol Pathol 79, 187 to 193, 2005. The UniProt Consortium. UniProt, the Universal Protein Knowledge Base. Nucleic Acids Res 46, 2699, 2018. Sklark-Zeik, D. et al. String V11. Protein-protein association networks with increased coverage, supporting functional discovery in genome-wide experimental datasets. Nucleic Acids Res 47, D607, D613, 2019. Aguet, F. et al. The GTEx Consortium Atlas of Genetic Regulatory Effects Across Human Tissues. Science 369, 1318 to 1330, 2020. Download references. Acknowledgements. RAS acknowledges funding through the European Union Horizon 2020 program Infrae Project Epic XS, project number 823,839, and the research program NWOTA with project number 741.018.201, which is partly financed by the Dutch Research Council, NWO. AE was funded by the Wetenschapsraad, grant number 2016-03798 and 2021-03979, and the Knut and Alice Wallenberg Foundation. The computation/data handling were enabled by the supercomputing resource Berzelius provided by the National Supercomputer Center at Linköping University and the Knut and Alice Wallenberg Foundation and SNCC, grant no SNCC 2021 5th-297 and Berzelius 2021-29. JEB acknowledges funding from the National Heart, Lung and Blood Institute, grant no. 5R35GM 13625, and the National Institute for General Medical Sciences, grant no. 5R01HL 144778. P. Beltrao is supported by the Helmut Horten Stiftung and the ETH Zurich Foundation. Funding. Open access funding provided by Stockholm University. Author information. Author notes. These authors contributed equally: David F. Burke, Patrick Bryant, Inigo Barrio-Hernandez, Danish Memon, and Gabriele Pozzati. Authors and affiliations. European Molecular Biology Laboratory, European Bioinformatics Institute, EMBL EBI, Cambridge, UK. David F. Burke, Inigo Barrio-Hernandez, Danish Memon, Alistair S. Dunham, and Pedro Beltrao. Science for Life Laboratory, Stockholm University, Solna, Sweden. Patrick Bryant, Gabriele Pozzati, Aditi Shenoy, Wensi Ju, Petras Kundrotas, and Arne Elofsson. Department of Biochemistry and Biophysics, Stockholm University, Stockholm, Sweden. Patrick Bryant, Gabriele Pozzati, Aditi Shenoy, Wensi Ju, Petras Kundrotas, and Arne Elofsson. Biomolecular Mass Spectrometry and Proteomics, Bijvoet Center for Biomolecular Research and Utrecht Institute of Pharmaceutical Sciences, Utrecht University, Utrecht, the Netherlands. Pascal Albanese and Richard O. Scheltema. Netherlands Proteomics Center, Utrecht, the Netherlands. Pascal Albanese and Richard O. Scheltema. Department of Genome Sciences, University of Washington, Seattle, Seattle, Washington, USA. Andrew Keller and James E. Bruce. Department of Biology, Institute of Molecular Systems Biology, ETH Zurich, Zurich, Switzerland. Alexander Leitner and Pedro Beltrao. Center for Computational Biology, the University of Kansas, Lawrence, Kansas, USA. Petras Kundrotas. Contributions. DFB analyzed disease-causing mutations in interfaces, phosphosites, and cross-linking with help from IBH, DM, ASD, and P. Beltrao. P. Bryant ran the prediction for the WHO-RE dataset. PA, AK, RAS, JEB, and AL provided data for the analysis. AE provided the prediction for the WHO.map dataset and analyzed the complex structural features with help from PK, GP, AS, P. Bryant, and WZP. Beltrao, and AE wrote the manuscript with help from all authors. Corresponding authors. Correspondence to Petras Kundrotas, Pedro Beltrao, or Arne Elofsson, towards a structurally resolved human-protein interaction network. David F. Burke, Patrick Bryant, Inigo Barrio-Hernandez, Danish Memon,
Gabriele Pozzati, Aditi Shenoy, Wensi Alistair S. Dunham, Pascal Albanese, Andrew Keller, Richard A. Scheltema, James E. Bruce, Alexander Leitner, Petras Kundrotas, Pedro Beltrao, and Arne Elofsson. Nature Structural and Molecular Biology, Volume 30, pages 216-225, 2023. Cite this article. Abstract. Cellular functions are governed by molecular machines that assemble through protein-protein interactions.